So here we are again back with a new video about the linkage and the crossing over, which is also part of the genetics. Now linkage and crossing over, these both are pretty much linked topics. If you understand one, you definitely get to the other one. Now in the context of examination, linkage has only been asked one time, which was in 2066, and crossing over, it is asked almost 5 times in the past 23 years. And when we are dealing about the medical examinations, the entrance examinations, one question will be fixed from this chapter. Every year one question is asked from this chapter, linkage and crossing over. So it is an important chapter, and yeah, though it is only asked one time, but it can be the probable question this year. So do not skip the topic. It is pretty much important topic and very easy topic. So first of all, head on to linkage. So what is linkage? By name we can understand linkage means the tendency of remaining together. So linkage, it is the tendency of two or more genes in a chromosome to remain together. For example, this is a chromosome. These both are the chromosomes. This is the sister chromatids, this is the centromere, okay, and these both are the sister chromatids. For example, this is the centromere, these both are the sister chromatids. So this is one chromatid, this is another chromatid. So these both are the sister chromatids of a chromosome. So we are drawing in somewhat this shape. So now see, here is one gene, here is another gene, okay, and here is another gene. Suppose here is a gene A and here is a gene B. So these both genes, suppose they are linked genes, we call it linked genes. Then what we mean by calling them linked genes is that when they are inherited to the offspring from the parents, these both characters are always linked. They both characters, they are not separated from one another. There is no chance of separation between these two characters. It means that if this character is there, then obviously in that offspring even the B character will be present. If A is not there, B will not be there, but if A is there, B is also there. This is the linkage. Now right now it might be confusing, but now I will give you the examples of the linkage, then you will understand this better. So in the examination, this question is asked somewhat this: what do you mean by linkage, and describe the types of linkages with the examples. And definitely only this question can be asked from this chapter, linkage. So first of all, what is a linkage? Linkage, it is the tendency of two or more than two genes to remain together when it is expressed from the parents to the offsprings. And the strength of linkage, it is determined by certain factors. Mostly it is determined by the distance between the two genes. If the distance between the two genes it is less, then the linkage strength will be more, and if the distance is more, then the linkage strength will be less. And there are other factors also, but that is not our concern right now. So now they will be asked, they'll be asking the types of the linkages. So basically there are two types of linkage. One is complete linkage, okay, and another one is incomplete linkage. So first of all, we'll be describing about the complete linkage. And if you are getting confused about these chromosomes and all the structures and all, forget this right now. That's not our concern right now. You'll be understanding about this very clearly in the crossing over. So first of all, complete linkage. Now complete linkage is a type of linkage where there is no chance of crossing over. Now for understanding linkage, we also need to understand the basic definition of crossing over first. In detail we will be reading after this, but here crossing over, for a glance, crossing over, it is a type of process in which there is exchange of the genetic materials between the paternal and the maternal chromosomes. If this is one chromosome and this is another chromosome, then these both chromosomes overlap with each other, and then there is exchange of the genetic materials and information between them, and then new combinations are produced. That is crossing over. We'll be reading about that later on, but for now you just need to understand that if there is crossing over, there will be appearance of a recombinant. It means there will be appearance of such characters, such individuals which are neither of the paternal type, neither of the maternal type, nor it will be of the father type, nor it will be of the mother type. They will be the mixture of them. Such mixtures, they are also called as crossovers and commonly known as recombinants. For now you just understand that if in the complete linkage there is no chance of crossing over, and since there is no chance of crossing over, there is no recombinant formed. Now no recombinant form means the offsprings which are produced in this type of link, in this type of crossing, they are all of the paternal types and the maternal types. It means that it is maintaining the parental trait from generation to generation. Now here we'll give the example of the complete linkage in Drosophila. Now for complete linkage we are taking the characters of the Drosophila, which are the wings and the eye color. Okay, so vestigial and normal wing and red and purple color, these are the characters which we will be taking. So now after writing just these informations, 2 to 3 lines information, we'll write the complete linkage in Drosophila, which will be the example. So now we are going on for the example for the complete linkage. For this example we are taking a Drosophila, the 2 Drosophilas which are of somewhat these types. One Drosophila is double dominant, red eyed and has normal wings. Okay, one parent is this which has red eyes and the normal wings, and the other parent, it has purple eyes, this is double recessive, purple eyes, and it has vestigial wings. So this is our double dominant parent, red eyed and normal wings, both are dominant, red is dominant and the normal wings are dominant. And this is a recessive parent which has both the characters recessive, purple eyes is recessive and the vestigial wings is recessive. So these are the phenotype of the parents. Now if you write the genotype of the parents, it will be red eyes will denote by capital R, okay, and we will be denoting the normal wings by capital W. So these are the 2 characters here. Then we have the characters purple eyes small r and vestigial wings small w. So these are the genotypes of the parents. Okay, and since we know that they occur in pair, so it will be RRWW, RRWW. Okay, now first of all the gametes are formed. We know that the gametes are formed. So first gamete formed will be this RW, another gamete will be RW. Again we have 2 gametes. So these are the gametes formed. Now we'll do the crossing. Now here in this chapter in the linkage we do not need to show the complete crossing, all the crosses and all. If we even show one progeny of the, it means only one F1 generation here, it will be enough for us. So we do not need to show all the crossing between them. You can directly give an arrow, because we know even if we cross all these, the result will only be the same. It will be this. All the F1 progenies will be same as this, because we know that in F1 every progeny it is hybrid and it is same as of Mendel's law. So these are our parents. Okay, these are the gametes, this is the genotype, and this is our F1 generation, which is hybrid, but again that is genotypically. But phenotypically if you see, again it will be red and it will be normal winged, because red is dominant and capital W, normal winged is dominant here. So this is our F1 generation. Till here we understood it was completely easy. And now here comes the complexity. Not that complexity, but the thing that we need to remember while showing the example of the linkage. What we need to do till F1 generation, after we show this, we need to cross the F1 generation with the recessive parent. It means we have to do the test cross. So as I said you about the test cross in the very first video of the genetics, the test cross is a type of cross in which the F1 generation, it is crossed with the recessive parent. So our F1 generation was this. Now we cross it with the recessive parent. Here the recessive parent is this. Okay, we cross with the recessive parent. Now see, again the gametes will be formed. Now here you need to remember one thing, that normally here 4 types of gamete would be formed. First I will show what 4 types it would be formed. If you see, normally there are 4 types of combinations that can occur here. If you remember the dihybrid cross of Mendel, it is similar to that. So capital R, capital W. Okay, capital R, small w. Then we have small r, capital W, and small r, small w. Normally these 4 gametes would be formed, but in this case of complete linkage, these 4 gametes are not formed. Only 2 out of them are formed. Now the two out of them which are formed, this is one type which is formed and this is another type which is formed. These both gametes are not formed. Now see why it is not formed. Normally, see, this is the same thing. Now only two types of gametes will form, which is capital R, capital W, small r, small w. Because we are dealing the case of complete linkage, it means we are dealing the case in which there is no crossing over. It means that the genes are not separated. Suppose these genes are lying on one chromosome. When I told you that the genes, they do not get separated in the linkage. So see, these both genes, capital R and capital W, they are lying on one chromosome, and we know that small r and small w, they are lying on another chromosome. Alternate pair lie on either of the chromosomes, isn't it? If this is the capital R, it means red, then this purple will be lying in the same locus but on another chromosome. Similarly, capital W and small w on alternate chromosomes. So basically, though they are lying on alternate chromosomes, but these both are lying on the same chromosome, capital R, capital W, and since we are dealing the case of linkage, they both will not separate, and they both will not separate. So if they both are not separating, how this combination will be formed? Capital R, they are not separating, so small w, in place of small w there should be capital W, since they both are not separating, but here is small w, so this case cannot form. Similarly, here see, capital W is there, but where is the capital R? No capital R, small r is there, so this case is not possible in this linkage. So only two types are formed, which are capital R, capital W, small r, small w, in which these both genes are not separated and lying together. Okay, back to the cross. So these are the gametes formed, and then this case here the gametes formed, simple, the gametes will be these. Okay, now what we do, the crossing one. Again, if you want to show the complete cross, you can, or if you just want to show the Punnett square, you can, but I'll show you. Now I'll just show you the result of the cross. After the result, capital R, capital W, so this will be our first, okay, then this, then this, and again this. Now you see the generation, this generation, it is same as of parent. RRWW, RRWW, same as parent. Capital R, capital R, small r, capital W, small w. It means hybrid, red and normal winged, same as of parent. So what we are seeing, two of them are same as of parent, and another two of them same as of another parent. So here no new combinations are formed. No such combination is formed which is different from the parental type. So this is the example of the complete linkage. In complete linkage, no such offspring is formed which is different from the parent. Again, I'll show you complete linkage from the beginning. First of all, I'll talk you in the context of examination. While writing in the examination, we'll just give the definition of the linkage, and then we'll write the complete linkage. It is a type of linkage in which no crossing over is there, so no recombinants are formed, only the paternal and the maternal types are formed. Example is complete linkage in Drosophila. So when we are dealing the complete linkage in Drosophila, we take the characters red-eyed normal winged and purple eyes vestigial wings. We'll write the genotype, and then gametes will be formed, crossing. These are the F1 generation. Till here it is pretty much simple. F1 generation is formed. Now what we do, you need to remember that the F1 generation need to be test crossed with the recessive parent. So this generation will cross it with this. Again, this and this is crossed, which is known as test cross. So on test cross, we need to remember that normally what four types of gametes we form according to the law of segregation of Mendel, those four types we are not making here. Only two types will be there, because I explained you these both genes, these both genes, they cannot separate from each other. So such gametes are not formed in which these genes are separated. Only the gametes are formed in which these two genes are present together, because it is a case of linkage, and that's what we are dealing. Linkage, the genes are linked, they are never gonna separate. So these two genes are linked. So finally, when we test cross, these types of characters are present. Now when you see these characters, you see that red normal, okay, red-eyed normal winged, again red-eyed normal winged, it is same as of this parent. Here purple-eyed vestigial winged, purple-eyed vestigial winged, same as of this parent. So you are seeing that the red color is always present with the normal winged, and purple color is always present with the vestigial winged. So we conclude that these two characters are completely linked with each other. It means if red color is there, then the normal wing will be present, and if purple color is there, vestigial wing will be present. And that's all about the complete linkage. These two links are So these two genes are linked together, and that's what we prove from the crosses. So now we have the another type, which is the final type, incomplete linkage. By name it must be clear that it is the alternate form of the complete linkage. It means that in complete linkage there was no crossing over, no recombinants, but here crossing over takes place. In such crossing over takes place and recombinants are formed, and recombinants are such progenies which are different from the parents. They are neither of the paternal type nor of the maternal type, they are the mixture of both of them. So due to wide space between the two genes, crossing over takes place, and when the crossing over takes place, both the paternal and the maternal genetic characters, they combine with each other and they form a character which is a mixture of both of them, which is known as recombinants. We'll be reading about the crossing over in detail after learning about the linkage. So here now we have the example of incomplete linkage. When we take the example of incomplete linkage, we take the example of the maize seed. Again, the thing what is need to be remembered here is the characters what we take in the crossing. So the two characters which we are taking here, the two characters are taken because we are dealing about linkage, so two genes must be there. So always we are taking two characters in both the complete and incomplete linkage. So the characters which we are taking here, they are first of all the double dominant one, colored and filled seeds. So this is the double dominant type. It means the endosperm we are using as double dominant is colored and it is filled seeds. Now filled seeds means fold scutellum. We'll be using this term, scutellum. Scutellum is a secretion, okay? So it is, the endosperm is colored and it has scutellum. It means filled seeds. But the recessive one which we'll be using, it will be colorless and shrunken seeds. Now shrunken seeds means it is empty scutellum. It does not have scutellum, so they are shrunken. Since scutellum is a fluidy substance, it is not present in the endosperm, then it is shrunken. So that these are the two types we are using. So as of complete linkage, we need to show the crossing over now again. So first of all, we need to write the parents. It means colored and full, and we have colorless and shrunken. So these are the phenotype. Now we'll write the genotype. Genotype means capital C will be there, double we'll write because they are present in pairs, and for this capital S, it means it contains the scutellum. Then we have for this small c, small s. Okay, these are the gametes. Sorry, this is the genotype. Now it will form the gametes. So gametes formed obviously two types, CS. Here also two types. Now we'll do the fertilization, and after the fertilization we obtain only one type of generation, which is this. This is the hybrid form, and hybrid form it is colored and filled because the dominant one is there, okay, and this is the hybrid one. Till now it is same as of complete linkage, but now on test cross, again we'll do test cross. On test cross we find the difference. We do the crossing of this F1 generation with the double recessive parent in test cross into this recessive parent, as we did in the complete linkage. Now the gametes formed will be of four types. Here we showed you in this test cross of the complete linkage that only two types of gametes were formed, and remember this reason you will not get in any book or any guide, not in your textbook, not in your answer book. This is created by Education Arena itself, after much research found out by me. So remember this carefully, because you will find this only in this video, that capital R, capital W and small r, small w are only present. Why these both are not present? You can get confused if you directly see the book. So this is important to understand why they are not present. I told you earlier that they are linked, so they do not get separated. Okay, this is a basic logic, but most of the students they get confused here. because they do not find the reason anywhere, and directly they are seeing two gametes. Then where are the other two gametes? We learned in the Mendel's law that four types of gametes will be formed here. So how only two types of gametes are there? And in the incomplete linkage again four types of gametes. So this can be pretty confusing, so I told you the reason as well. Here only two types were there. Now here according to the law four types of gametes will be formed. Okay, so capital C capital S will be first one, dihybrid cross, similar to dihybrid cross we did in Mendel 1. Capital C small s another type, capital C small c capital S another, small c small s another. And here only two types will be formed. Okay, so now if we do the crossing, here the crossing will be pretty much different because many combinations can be formed. So see, first combination will be this. Okay, so it is the same combination will formed if we cross this with this and this with this. So let us cancel this. It will be just a confusing thing. Actually the generations will be same, but here we are not showing it, otherwise it will be like a mess here. So capital C capital S small c small s. So this is the first type. Another type we are seeing, okay, then third type, and the fourth type. Now see the generation. Capital C small c capital S small s, same as of parent. Okay, done. Small c small s same as of parent, done. But these two characters, capital C small c, okay, but again small s neither of the parent is matching. Even here capital S small s is matching, but small c is of this. So these two characters, they are not matching from either of the parent, and these two characters, these two progenies which are formed, these are known as the recombinants, or they are known as the crossovers. These are formed as a result of crossing over. We'll be learning crossing over after this. So these two are the recombinants which are formed in the incomplete linkage. These recombinants, they are not formed in the complete linkage, they are only formed in the incomplete linkage. Okay, so the ratio, if you see, it is that two of them, they are of paternal types, and two of them they are of not paternal. One is paternal, one is maternal. Two of them are parental types, and two of them are recombinants, which are neither of the parental types. So this is all about the linkage. Once again from the beginning, I'll tell you linkage definition in the context of examination, then you write about the description of the complete linkage, and then you have to write about the example. You can show the crosses first, and then you can write the description of the crosses. Description is based on the cross itself, pretty much simple. These two are crossed and gametes are formed, and then in F1 generation, this on test cross we find that these characters are formed, only two gametes. This information, it's your wish, you can give or not, because most of the times it is not given. So these are the four types, no recombinants, but in incomplete linkage even the recombinants are formed. Till here it is same, only here the four types of gametes are formed, and in complete no four types of gametes are formed, only two types of gametes are formed. So this is all about the linkage. Now one thing, do not get confused between the previous videos and this. You can get confused which characters are used where. You might mess up in your mind, but it's pretty much simple. In the linkage, you just need to remember that the complete linkage is of red-eyed normal wings, incomplete is of the maize seed colored and filled scutellum, and then we were using the sex-linked inheritance in the previous video. There the traits we were using, it was the red-eyed female with the white-eyed male, and then the hybrid female with the white-eyed male, and then again the red male with the white female. It was about the eye color. Okay, so if you see the previous video, you will again be clear about it. So in that video we were discussing about the eye color. Here we are discussing about the eye color as well as the wings. In this way you can compare all the crosses, and then you can remember them. Just do not get confused between the crosses. The only thing you need to remember is linkage is that you just have to do the test cross after the completion of the cross till F1 generation. You do till F1 generation and you do the test cross F1 generation with the recessive parent. That's all you need to remember in the linkage. So now we have our next topic, which is the crossing over, and this is much more important than linkage because it is asked more times than linkage, and the probability of asking crossing over is much more than the linkage. So first of all, what is crossing over? So crossing over, it is a process of exchange of the genetic materials between the non-sister chromatids of the homologous chromosomes. Now here I use the term non-sister chromatids of the homologous chromosomes. What do we mean by that? So first of all, I'll give you the basic knowledge of the chromosomes. This is the centromere, okay? This is one chromatid, this is another chromatid. So the two chromatids, they are joined by centromere. This is one chromatid, this is another chromatid. These two are joined by a centromere. So this is joined by a centromere to this. So this is one chromosome. Suppose this is the paternal type from father. Then we have a maternal type for this also, okay? So again, one chromatid. This is one chromatid, completely one chromatid. Here is centromere. And then we have another chromatid. So two chromatids of this, two chromatids of this. This and this are sister chromatids. These both, the two chromatids, are known as sister chromatids. Even they are known as sister chromatids. They both are homologous chromosomes. Homologous chromosomes means the pair of chromosomes, one from the paternal, one from the maternal. So these both are homologous chromosomes. They both are sister chromatids. But see, one chromatid of this and one chromatid of this, they are non-sister chromatids. The chromatids of a same chromosome, they are the sister chromatids, but the two chromatids of the two different chromosomes, one from paternal, one from maternal, these both are the non-sister chromatids. So basically, crossing over is a process which is done between these both non-sister chromatids. From the definition, we came to know that it is a process of exchange of the genetic materials between non-sister chromatids of the homologous chromosomes. So these are the homologous chromosomes. The process of exchange of the genetic material between these two non-sister chromatids, it is the crossing over. So basically, how this crossing over occurs, that is our concern, and that is what will be asked in the examination. We will be asked to describe the process of crossing over. Okay, we will be told to describe the mechanism of the crossing over. If you see your set book, you will find this question pretty much as the mechanism of crossing over. You will find this question asked so many times in the examinations. Okay. So what is the result of this crossing over? After this crossing over happens, one gamete, it is of the paternal type, one gamete, it is of the maternal type, but these two gametes, we are saying that these two chromatids, these two chromatids, they interchange the information, and when interchange of information is there, exchange of information is there, then this chromosome, it consists of a mixture of paternal and maternal. Even this consists of a mixture of paternal and maternal. So these both are different from the paternal and maternal. So in this way, four types are formed, in which two are the paternal and maternal parental types and two are recombinants. So actually, the progenies which are formed as a result of crossing over, they are the recombinants. So crossing over, it helps in the production of recombinants which are different from the parental types. So now we are going on for the mechanism, actually how it occurs. So basically, this crossing over, it occurs during the prophase 1 of the meiosis division. The meiosis cell division, it has 5 stages, which are the prophase, metaphase, anaphase, and so on. So right now we are seeing that in this meiosis cell division, actually in prophase 1, there are further 5 stages, which are leptotene, zygotene, then pachytene, then we have diplotin, and then diakinesis. So these are all the things we do not need to remember right now. I am just telling you for information. In this meiosis cell division, in the prophase 1, this crossing over takes place. Basically in pachytene, to be precise, in zygotene and pachytene, it takes place. So during this stage of the cell division, crossing over takes place, and the points in which the crossing over takes place, they are known as chiasma. There can be one or more than one chiasma in a process. At one place, exchange of information can take place, or more than one place there can be chiasmas. So now let's move on for the mechanism of the crossing over. So we can define the mechanism in 4 simple steps. Now believe me, crossing over is a very simple process. It is not very complex as of mechanism of DNA replication. It is very simple. We can define it in 4 steps. First of all, we have synapsis, okay. Then we have duplication of the chromosomes. Then we have the crossing over for precisement, and then we have the terminalization. So we'll be defining each of the steps in detail. So first we have the synapsis. So what is exactly synapsis? What happens is that in the first step of crossing over, during the zygotine stage of the prophase 1, I told you that prophase 1 has 5 substages: leptotin, zygotine, pachytin, diplotin, dikinesis. Here the thing you need to remember is zygotine and pachytin. Only 2 names you need to remember, okay? Zygotine, pachytin. So what happens is that in the zygotine stage of the prophase 1 of meiosis cell division, the 2 homologous chromosomes, they just come close to each other, okay, and they form a pair which is known as bivalent. Now actually what happens is that the chromosomes here in this stage, in zygotine stage, as I told you that there are 2 sister chromatids, but in this zygotine stage these 2 sister chromatids are not clear. It is these 2 sister chromatids, they are so close to each other that they seem as only one chromosome. It means only one chromatid, okay. These both are so close to each other that we can see it as only one chromatid. So what happens is that in zygotine stage, one chromosome, now though it contains the chromatids, this is one chromatid, here is centromere, these are 2 chromatids here, okay. These 2 chromatids are there, but in this zygotine stage it is shown as one. So what happens is that this one chromosome, it comes to another chromosome. This is the parental chromosome, and this is the paternal chromosome. This is the maternal chromosome. These both chromosomes, these both homologous chromosomes, they come close to each other and they form a pair which is known as bivalent, okay. So these both chromosomes, they come close to each other and form a pair which is bivalent. Since there are 2 chromosomes, so it is known as bivalent, and this process is known as synapsis, and that is all about synapsis. Just 3 lines to write that it happens in the zygotine stage of the prophase 1 of meiosis, and the homologous chromosomes, they come close to each other to form a pair. The pair is known as bivalent, and this process is known as synapsis. Then we have duplication of the chromosomes. So duplication of chromosomes is also as short as of synapsis. This is just a process in which what happens is that this first of all happens in the pachytine stage, okay. Now from zygotine we moved on to pachytine. So in this pachytine stage, what happens is that these 2 bivalents, this one bivalent actually, this one bivalent we are seeing, the 2 non-sister chromatids which are present here very close to each other, now they both differentiate. It means that each chromosome of the homologous pair, it differentiates into 2 sister chromatids. So this is just a single chromosome right now, and it differentiates into this 2 sister chromatids. So it was somehow like this, but now what happens is that it becomes, this is one chromatid, this is another chromatid. Centromere is there, one chromatid, another chromatid. So in this way, this bivalent, it formed into tetravalent. Now why it is tetravalent? Because see, we are seeing 4 chromatids are there. Now basically in terms it is known as tetrad. So you must have heard the term tetrad formation. This is the tetrad formation. Just the thing is that the bivalent, which had the 2 chromosomes in it, now the 2 chromosomes, they differentiated into 2 more sister chromatids, and in this way 4 chromatids are there. So it means tetravalent is there, so it is known as tetrad. Okay, so that's all about the duplication of chromosomes, and so it is known as duplication of the chromosomes. Each chromosome is duplicated into 2 sister chromatids. Okay, still it is close to each other, okay, which happened in the synapsis. Both are close to each other, so pairing is there. This is the pairing. 2 homologous chromosomes are there. Even here homologous chromosomes are there. They are paired together, very close to each other. Now we have the 3rd step, and the 3rd step is crossing over. Till now it is very simple. Even now it is very simple. Crossing over, what happens is that this 2 sister chromatids, these two sister chromatids and these two sister chromatids are there, but between these two non-sister chromatids which are paired together, these both non-sister chromatids which are paired together of the homologous chromosome, they start twisting over each other. It starts twisting over each other and it starts twisting over each other. Okay, so what happens is that when twisting over takes place, it is due to the action of an enzyme known as endonuclease. Okay, so this twisting over takes place and it takes place at a point known as chiasma. So this point is known as chiasma, even this point is known as chiasma, and due to this twisting, the genetic materials, they get exchanged. So this is the homologous chromosome. Okay, so what happens is that these two non-sister chromatids, they start twisting over each other, and when they start twisting over each other, they exchange genetic materials between them, and they start twisting over at places known as chiasma. So this is a pretty simple thing, but I'll show you in more detail about this in the context of genes, by showing you genes how they exchange. So see, first of all, I'll show you diagrammatically. Diagrammatically, two sister chromatids are shown this way. Okay, this is the centromere. Actually, it is in this shape, but diagrammatically we are showing in this shape. This is the centromere. These are the two sister chromatids. Suppose this is containing a gene A, gene B, gene B. Okay, these two are linked genes. See, I told you that genes are present in pair. In this figure, you'll understand each and everything, that the genes are present in pair and then the alternate and all. Okay, right now this is out of our topic. I'm telling you out of the topic. If you want to draw this figure, you can or not. So see, this is just extra information. So this is one chromosome. Suppose this is the paternal chromosome. Okay, it consists of gene A and gene B. We know that genes are present in pairs, so we write somewhat this way. Okay, so this is a pair, this is a pair. These both are linked genes. A and B are linked genes. This is the recessive one, so we are writing in small. Now we have the maternal one also. These are the two sister chromatids of the maternal one. Okay, now this contains capital A, capital A, capital B, capital B. So again, these both are linked genes. So now see, you have that this is small a, its alternative form is there, capital A, the dominant one. Even here, dominant one, recessive one, and A and B are linked genes. Now what happens is that this is the case of the duplication of the chromosomes, synapsis and duplication of chromosomes. This is the structure of first and second case, these both cases. Okay, to be precise, this is the case of duplication of chromosomes, because two sister chromatids, they are clearly seen. Now what happens is that in this third step of crossing over, as I told you that at point chiasma, they start twisting over each other. What happens, to be precise, is that this is a centromere. Again, I'll draw this figure. So these are the two non-sister chromatids which are paired with each other. Okay, so what actually happens is that these both start pairing over each other. So this pairs over there and this pairs over here. In this way, they start pairing over each other. It means A gene was presented, B gene was A, B. Okay, so this is right now easy. But this B gene which was present here, it comes here, and the capital B which was present here, it goes there. Capital A goes there, capital B. Okay, in this way we have four gametes, A, B. Okay, capital A, capital B. Now where here we have small a, capital B, small a, sorry, capital A, small a. In this way, four types of gametes are formed, in which two are parental types, but these two are recombinants. So in this way, crossing over helps in the incomplete linkage. Okay, finally, if you see, after the terminalization, I'll tell you about terminalization, but here after terminalization we'll find the chromosomes to be this way. So this will be containing the same gene A, B. This will be containing the same gene capital A, capital B. But here the genes will be different. Capital A and the capital B of this, it twisted and went there. So capital B. Again the small b of this, it twisted and went here. Twisted and went here. So capital A and small b. So these will be the types of the genes which will be formed after the crossing over. You can see two recombinants are formed which are different from the parents. So that is about the crossing over. Now back to the topic in the context of examination. Till here I told you that in crossing over at points chiasma, just what happens is that the twisting between the non-sister chromatid, it takes place, and in this way the genetic materials, they get exchanged between the homologous chromosomes. So after writing these 5-6 lines, we'll write the final step, which is our terminalization. So by the name it is clear that it is the final step, terminalization. So what happens is, on the completion of this crossing over, the non-sister chromatids, they move away from one another due to the repulsion between them. It means these two chromatids which were crossed, which were very close to each other, non-sister chromatids, they start moving away from each other. Okay, so what happens is that this chromosome, it moves away, and again they both start repelling each other, so they both move away from each other. In this way, the terminalization is there, they both get repelled from each other. Now basically this repulsion, it starts from the centromere. So how this repulsion starts, these are the paired chromosomes. Okay, now this is the stage when the crossing over is complete. Now these are the chiasmas, we know. So these are the centromere. So basically what happens is that the suppose the chiasma can be formed even here the chiasma can be there. I told you as many chiasmas as possible can be there. So what happens is that from the centromere, the chiasma start moving towards the end. Suppose here is one chiasma, then it starts moving towards the end. I'll show you in the figure again. Suppose this gene, this non-sister chromatid, this is the chromosome. In this way, these both are one chromatids, and these both are the two paired chromatids. So in this way, one chiasma is here, another here. Okay, in this way, these are the places where the genetic information is exchanging, I told you, in the crossing over. So what happens is that in terminalization, from the place of centromere, it means from the center, these chiasma start moving towards the end. So this chiasma, it disappears and it starts moving towards the end. So this moving of chiasma towards the ends, to the terminal ends of the chromosomes, it is known as terminalization, and on that basis the step is given the name terminalization. Okay, so this process, it involves the movement of the chiasma towards the end of the chromosome, and in this way, since it is moving towards the end of the chromosome, and finally they both separate from each other. So that's all about the terminalization, and that's all about the crossing over. Once again, from the beginning I'm telling you, so that you do not get confused, we have 4 steps. Synapsis. In the synapsis, the two chromosomes, they come close to each other. For now, they are not differentiated into sister chromatids, so they are known as bivalent. In the duplication of chromosomes, they separate into sister chromatids, so they are known as tetrad. Now in crossing over, what happens, that the two non-sister chromatids, they twist over each other at the places known as chiasma. There can be more than one chiasma. So in this way, the exchange of information takes place. As a result, 2 out of the 4 chromatids, they are of paternal types, and another 2 out of 4, they are recombinant types. And finally, terminalization, the chiasma which is formed, it starts moving towards the end, and finally the chiasma disappears. It means that due to the repulsion between the two non-sister chromatids, the two chromosomes, they get separated, and in this way the crossing over ends. It was very simple, isn't it? So out of 4 gametes after meiosis, 2 parental combinations are there with 2 new combinations or recombinants, which is produced as a result of crossing over. So that's even the case of incomplete linkage. In incomplete linkage, we told you that crossing over is there in incomplete linkage, and since crossing over is the recombinant is formed, so in this way, in detail, we can see that in this way crossing over was there and recombinant was formed. Okay, so finally it's over, but sometimes even the significance of the crossing over may be asked. So 2-3 points about that. So crossing over, basically the significance is that it produces the recombinants. Okay, now recombinants are produced, it means that the new combinations of the genes are produced. This is one significance. Another significance, it can be that new recombinations which are produced, it can be used by the plant breeders plant breeders usually they use new combinations to produce new types of plants. Okay, and the third importance, it can be the adaptability. Okay, you think that our ancestors would be same as us, then we would not be able to adapt outside to the changing world. If we would be same as our ancestors, we could not adapt to this changing environment. So in this way, this recombination, it separates the offsprings from the parents, and it provides a space for the adaptability. It helps in adaptability. This crossing over, it is helping in adaptability because it is separating the offspring from the parents. We are different from our ancestors, so we are adapted to this environment. Our ancestors could not adapt to this environment, so it helps in adaptability and speciation. So that's all about the crossing over, and with this we end the chapter linkage and crossing over. For you, if you didn't understand the terminalization, because terminalization is quite confusing, as I showed you, I have a video for you, the movement of the chiasma towards the end. After seeing this video, you can understand terminalization much better. And that's all for today. Wait till the next video. Take care. Goodbye.